Hello, today I'm gonna present this paper, Adhesion and Formation of Microbial Biofilms in Complex Microfluidic Device. For the introduction, we need to know that Shewanella oneidensis is a metal reducing bacteria, which is of interest of bioremediation and clean energy application. In this paper, they studied the spatial temporal dynamics of biofilms, such as spatial distribution, cluster formation, and surface coverage. They compare higher flow rates and lower flow rates and how it participates in the biofilm formation. For this study, they used the atomic form microscopy, AFM, for observe the production of extracellular polymeric substrate, EPS, that occur at a higher flow velocities. It's important for the introduction to know that biofilms are aggregation of microbes adhere in a surface, which are produced in a natural or artificial way. For analysis, this formation, they apply a microfluidic device. In conjunction with another technologies, which are microscopy, for investigate factors such as fluid dynamics. So in this paper, they quantify the effect of flow through a baffle structure of biofilm development of the bacteria using AP fluorescent microscopy, developing a deeper understanding of biofilm characteristics such as a distribution of extracellular polymeric substance, EPS, with atomic force microscopy. Here we can observe how is the design of this microfluidic device. It contains one outlet. It is made of PDMS and it has a cover sleeve. So for the inlet, it introduced the flow rate, and in the inside of the micro channel, it has these baffles that are these baffles are inside the micro channel and are inclined. We can observe here in the direction of the fluid flow. The baffle area is 2,500 micrometers in the length. So here when they apply the microscopy for observe how is the flows of these baffles, they have different flow rates. So the velocity contours of the fluid flow in the baffle regions, we can observe here. The color indicate the velocity magnitude. The color red corresponding to regions of higher velocity, and the blue corresponding to regions of lower velocity, like this. For the experiment, they use the bacterial culture and incubation, the Shewanella oneidensis. They inoculate in a LV broad in a shaking incubator at 30 grades centigrade. The microfluidic device has a width of 450 micrometers and a height of 125 micrometers, and contains a region with a periodic array of baffles that I indicated previously. So the inoculum was infused into the channels by means of a syringe pump, and they prove here two different flow rates, one of 8 microliters per hour and another one 8 per 10 at -1 microliters per hour. Then they observe this fluorescent image of biofilm formation that using this program says Axioplan. For analyzing all these pictures, they using the image processing in my MATLAB. So here we can observe these two images that are taken in two different times at 0 hours and 3 hours. The average grade scale value per unit are also increased by an order of the magnitude. Here we can observe that the location with a brighter spot can be seen, which correspond to region of higher density. Here we have more density of microorganisms. These patched bright spots are also indicate the microbial growth. We can observe that here we have more spots of bright spots that in this picture that was taken at the first hours of flow. Microbial growth initiates at all the localization of the microchannels. We can observe in this picture that the final microbial concentration differs significantly from the initial profile. The microbial density is approximately equal at the all localization of the channels. And we can observe that it's very different at the first hours than the rest of the hours. With increased flow rate of the inoculum, the patterns and the dynamic of the biofilm seem different. Another important aspect that they analyze that is so interesting is that in a lower flow rates, they can observe an intact flagella in several of the microbes could be easily discerned. Compared with the higher flow rates, the flagella could not be easily discerned. For that, they conclude that the EPS production is response of the microbes to the higher flow rate conditions and appear to be a natural mechanism to allow microbes to adhere more firmly to the substrate. So cellular aggregation was compact at higher flow rates as compared at lower flow rate conditions. For this, the fluid flow rate plays a very important role in the microbial transportation and affect the biofilm formation. Here we can observe this image when they applied FNM for observe, and the flagella, they can observe intact flagella in the microbes at the low flow rates in this case. And when they compare the flow rates with the surface coverage, they have more surface coverage in a higher flow rate, like we can observe here in this graphic. In conclusion, the effect of hydrodynamic flows on biofilm formation is so important because here we can observe the behavior of the bacterium. In this study, they conclude that a higher flow rate is better for the formation of the biofilm. Thank you.